In alpha thalassemia, all of the normal adult hemoglobins have alpha chains. Okay, so our hemoglobin A has alpha beta, hemoglobin A2 has alpha delta, hemoglobin F has alpha gamma. And essentially there are 4 genes that determine the rate of synthesis of these alpha globin chains. And a deletion of any one of these 4 or a combination will lead to different severities of alpha thalassemia. So when I say that there's 4 genes, a child will get 1 pair or 2 genes from mom and then 1 pair or 2 genes from dad. Okay. So again, our alpha and our beta hemoglobin chains will pair up 1 to 1. But if we have a decrease in our alpha, that means that we'll have an excess of betas and an excess of gammas. So not an increased rate, just an excess. So maybe that's not the best. Shouldn't use arrows. Excess of betas and gammas. So those excess beta and gamma chains, they'll actually form tetramers. And there's tetramers of betas. And these are actually referred to as hemoglobin H. And then there's tetramers of gammas, which look like a little upside down fish. That's my attempt at that. So the 4 gammas, I'll write it down here. Those are called hemoglobin Barts. Okay. And some of these they'll precipitate and form inclusions in the cell. And then as the cells with those inclusions go through the spleen and the extravascular organs, specifically spleen, it leads to extravascular hemolysis. So not only do we have a decrease in our normal hemoglobin synthesis, but now we also have extravascular hemolysis, which both of those together leads to a lot of problems. So this is the inheritance combinations that you can see with alpha thalassemias. And the alpha thalassemia or alpha chain is on chromosome 16. And remember, you get 2 alphas from mom on 1 gene and 2 alphas from dad on 1 gene. Okay, and so here's the loci on 1 gene. So let's start over here with our silent carrier. This is just a deletion on 1 of the 2 genes. So you still have 3 functioning alpha gene loci. Okay, alpha thalassemia disease, also called alpha thalassemia trait or alpha thalassemia minor. These are 2 deletions, and it can be 1 deletion on each chromosome or 2 deletions on 1 chromosome. So either of those would be considered alpha thalassemia minor or disease or trait. Then we have hemoglobin H disease, which is 3 deletions. And then we have I'm going to take this part out right now. Then we have hydrops fatalis, which is a deletion of all 4 alpha chain genes. Here's a picture of those tetramer inclusions. And just as a reminder, tetramers are going to be 4 globin chains that are either beta or gamma. So 4 betas or 4 gammas. And to me, they look like golf balls. But these are done on or seen on a brilliant crystal blue. And this particular picture is of hemoglobin H inclusions, which are going to be the 4 betas. 4 gammas is referred to again as hemoglobin Barts. Okay, so here we're going to talk about our silent carriers. This is an alpha thalassemia. It is the least severe of the alpha thalassemias. There is only 1 deletion. So they have 3 functioning alpha globin chain loci. So hematologically, patients look fairly normal, but you may see some target cells and you may see some elliptocytes. Remember, target cells are very commonly seen in any disease that has something wrong with the hemoglobin itself. Really, the only way to diagnose this is with genetic testing. Then we have alpha thalassemia trait, or according to that one picture, also called disease, also called alpha thalassemia minor. Lots of names for everything. This is still not as severe as some of the other alpha thalassemias. We still have 2 viable alpha genes. So we have 2 deletions, and it can be on, again, the same gene or a deletion on both genes. So in alpha thalassemia trait, minor or disease, however you want to say that, some things that you're going to see, you could see a small amount of hemoglobin BARTs, and remember this is the tetramers of gamma, and it's going to be in a small amount because, I'm going to say this a couple different times hopefully, alpha thalassemia trait, a patient can be an adult with alpha thalassemia trait, okay? And we know that as an adult, the gamma is produced in smaller, we'll just say smaller production in adults. So that's why we'd only have 5 to 10% of those tetramers of gamma, okay? However, if it's found in an adult, these tetramers, this is diagnostic of alpha thalassemia. And I think this would probably end up being on an exam. Oh, and I do want to add, this diagnosis or the seeing the gamma tetramers, this does not differentiate the different types. So we'll just say not differential. It can just tell us that that patient has some form of alpha thalassemia. So what are we going to see in the lab? We're going to see mild anemia. We're going to see microcytic hypochromic cells. Do not confuse. We're going to say don't confuse with iron deficiency anemia. Oh, that's what the A is. Okay, don't confuse it with iron deficiency anemia. You're also going to see target cells and elliptocytes. So if we compare a silent carrier to now alpha thalassemia trait, the silent carrier also has targets and elliptocytes, but now we also have those microcytic hypochromic cells and that mild anemia. Then we have hemoglobin H disease, which is more commonly seen in Asian heritage. It is a pretty severe condition. There is only 1 functioning alpha gene, so 3 deletions, 1 functional. So now we're going to have both sets of tetramers. We're going to have our hemoglobin H, which is our beta tetramers. And we're going to have Bart's hemoglobin, which is our gamma tetramers. I don't know, tetramers. We'll also see A2, and then the rest of our hemoglobin will be A. So there's still alpha chains being made that are pairing with the beta chains. There's just not a lot. Clinical symptoms, there is some variation in how these symptoms are expressed, but there is some mild to severe anemia. I didn't write this down. So oxidative drugs can cause hemolysis, a hemolytic crisis in these patients. Oh, I did write it down. Splenomegaly, so you're going to have an enlarged spleen, bone changes, and lifelong anemia. And then transfusions are only given as needed. So in the lab, you're going to find that mild to moderate anemia, so something less than 10 grams per deciliter. Your retics are going to be increased. And then in the next slide, I'll talk about the smear. Okay, so on our smear, this is where things get kind of crazy. We're still going to have those microcytic hypochromic cells. So if you have a smear that's microhypo, again, it's going to either be a thalassemia or an iron deficiency anemia. Okay, so we do need to eliminate that possibility. All right, target cells. We're going to see poikilocytosis, all sorts of weird shapes, but not necessarily, I mean, other than targets and maybe some schistocytes, not necessarily any others that are very prominent. Okay, so we'll have that anisocytosis a little bit because our body is trying to spit out retics. Okay, but then we have also those microcytic cells, schistocytes, and that has to do with the extravascular hemolysis. Basophilic stippling because of increased erythropoiesis. And then we'll see those hemoglobin H or beta tetramers that are precipitated in those red cells. And again, this is going to be seen on a brilliant crescent blue. It's going to have that pitted golf ball appearance, as you see in the picture over here. And then just because it's good to hear a million things or hear things a million times, those hemoglobin H inclusions or those beta tetramers, they will cause a decrease in the red cell lifespan because the spleen's taking it out. Okay, it's trying to take out those inclusions. I do want to come back up here to this here, eliminating iron deficiency anemia as a possibility. So I do want you guys to pause for just a moment and think about what tests we can do, what a good test to do to determine if this is iron deficiency anemia or if it's a thalassemia. So pause here and think about it. And I paused, but you guys don't know that. So I was paused for all of like 30 seconds. So a good test that you can do is a ferritin level. In iron deficiency anemia, ferritin is going to be decreased. And in thalassemia, it's going to be normal to increased because there's not a problem with getting and keeping iron. So then our last one is hydrops fatalis. This is the most severe. There are no alpha chains being made because all 4 of the genes have been deleted. So there's no hemoglobin A being formed. There's no hemoglobin A2 being formed. There's not even hemoglobin F being formed. Okay, so this is made up mostly of hemoglobin BARTs, which are your gamma tetramers. There could be some hemoglobin H tetramers or your beta chains, but the reason that there might not be is because this is usually, patients usually die soon after birth, if not before birth. So the body hasn't had a chance to make any or much of those beta chains. So both of these hemoglobin BARTs and hemoglobin H, they have a high oxygen affinity, which means that they do not want to give oxygen up to the tissues. Okay, so they resist delivering oxygen to the tissues. So because of that, and this is awful to think about, but the baby basically suffocates prior to or shortly after birth because oxygen is not being delivered to any of the tissues. Okay, so it could be a spontaneous abortion, stillbirth, infant death within hours. Now, if a baby does make it to birth, not birth, third trimester. So if baby makes it to third trimester, what's helping it get there is hemoglobin Portland, Portland, which are chains, zeta chains and gamma chains. Okay. However, hemoglobin Portland cannot support later stages of fetal growth. So usually these births end, or these babies' lives end before birth, but once in a while you'll have a live birth that doesn't last. And then the last thing is that hydrops fatalis can be hazardous to mom, and that's due to toxemia. So laboratory findings, testing is done on cord blood, and it's usually post-mortem. So after baby has died, you're going to see that those microcytic hypochromic red cells, aniso and poikilocytosis, nucleated red blood cells. And I do want to remind you that newborns normally have, they normally have an increase in retics, but here there's going to be a marked increase. So let's say marked increase. And then you may see some sickling of those red blood cells due to the hemoglobin BARTs. On electrophoresis, there's going to be a real high percentage of hemoglobin BARTs.